Good day, biochemistry students. Today, we're going to continue our discussion, and this is part 3 of lesson 2. So, let's talk about the cyclic forms of monosaccharides and the Haworth projection formula. First of all, what do you mean by the cyclic forms of monosaccharides? This means that monosaccharides that contain 5 or more carbon atoms can form cyclic structures in aqueous solution. 2 cyclic stereoisomers can form from each straight-chain monosaccharides, and these are known as anomers. In our example, here we have 2 forms of D-glucose, the alpha form and the beta form. So it's right to say that alpha form and beta form are anomers of D-glucose. So here is one question. Why do monosaccharides form cyclic structures? Monosaccharides form cyclic structures because of the ability of the OH group and an aldehyde or keto group to react intermolecularly to form a hemiacetal or hemiketal formation. Remember, we have the terms hemiacetal or hemiketal. Hemiacetal is formed with the presence of an aldehyde functional group, while hemiketal is formed with the presence of a ketone functional group. So, look at our example here. The first one, we have the alpha form. So you can see OH of carbon 1. Okay, so let me just put a mark on that. You have the OH of carbon 1, right? And the CH2OH, this one, of carbon 5 are on opposite sides. So if you will count from here, this is carbon 1, 2, 3, 4, and 5. So the alpha form is formed when the OH of carbon 1 and CH2OH of carbon 5 are on opposite sides. Now, how about the beta form? The beta form, we have the OH, okay, we have the OH of carbon 1 and CH2OH of carbon 5, this one, are on the same sides. Now let's count the carbons. 1, 2, 3, 4, and 5. Okay? So how would you know that they are on opposite sides or on the same sides? Okay. So look at the direction of the OH in this one and the direction of CH2OH in alpha form. The OH is positioned downward, while the CH2OH is positioned upward. And that is why in alpha form, it says here that they are pointing on opposite sides. Okay, now look at the beta form. The OH is positioned upwards, and the CH2OH is also positioned upwards. And that is why in beta form, it has been mentioned here that they are pointing on the same side. Did you get it? I hope you do. It's easy, right? Haworth projection formulas. Haworth projection formulas are as useful as the Fisher projection. The Haworth projection is a way around this limitation that does not require you to try to convey the complete 3D image of the molecule. Sugars in Haworth projection can be classified according to the ring size, or the 5 furanosis or 6 pyranoses, which they assume in solution. A sugar with fewer than 5 carbons cannot form a stable ring. So, what is the importance of Haworth projection? Haworth projections are typically used to depict cyclic sugars. Sugars form a 6-membered ring, and they have several stereocenters. In order to distinguish between types of sugars, it is important to have a simple way to draw the sugar molecules while keeping the orientation of the OH attachment. In this example, we are shown the example of pyranose structure of glucose and the furanose structure of fructose and ribose. Let us focus on the pyranose first. Right. A pyran contains 6-membered ring. However, it only contains 5 carbons along with 1 oxygen. Right? So we have here our D-glucose. Let's count the carbon atoms. 1, 2, 3, 4, and 5. And the 6th member is an oxygen atom. That is why I have mentioned earlier that pyranose is a 6-membered ring, 5 of which is carbon atoms and 1 oxygen atom. Now look at the furanose. Okay, so furanose only contains 5 members, or it is a 5-membered ring. Let's count the number of carbon atoms. We have 1, 2, 3, and 4. And how about this one? 1, 2, 3, and 4. There are only 5 carbon atoms in furanose, and the 5th member is an oxygen atom. So remember, a pyranose is a 6-membered ring. A furanose is a 5-membered ring, right? But it has only 5 carbon atoms in a pyranose structure and 4 carbon atoms in a furanose structure. So what is the 6th member in a pyranose? It is an oxygen atom. And the 5th member of a furanose, that is the oxygen atom as well. Did you get it? I hope you understood the difference. It's easy, right? Now, let's proceed to the next. Now, the Howard projections are divided into 2 classes, as what I have mentioned in the previous slide. We have the pyranose and the furanose. Furanose are a 5-membered ring hemiacetals, wherein the oxygen are drawn at the top of a pentagon. The horizontal bond at the bottom is assumed to be coming out of the plane towards you, or that means towards the viewer. Thus, the 5-membered ring is in a plane perpendicular to the page. The vertical lines, on the other hand, are drawn in each carbon to indicate attachments above and below the plane of the 5-membered ring. In solution, the fructose, ribose, and the oxoribose will exist as 5-membered furanose rings. The furanose ring resembles the cyclic ether called furan. A furanose form of the sugar ribose is a good example. As what I have mentioned earlier, furanoses contains 4 carbons, and the 5th member is an oxygen atom. Okay, so 4 carbon atoms plus 1 atom of oxygen makes up a furan ring. Did you get it? Now, let's proceed. On the other hand, we have a pyranose, and it is known to have 6 members, right? In my example earlier, we have 5 carbons in a pyranose, and the 5th member is an oxygen atom. So 5 carbons plus 1 atom of oxygen makes it 6-member, or a pyranose. Pyranoses have a slightly different but quite similar Howard projection. So a hexagon is placed so that one horizontal bond runs along with the bottom. The oxygen in the ring is placed at the upper right. Okay. The oxygen in the ring is placed at the upper right. Usually, the hemiacetal carbon is placed at the extreme right. In solution, the glucose, galactose, and mannose will exist as 6-member pyranose ring. In the sugar ring resembles a cyclic ether called pyran. Again, the bond at the bottom is assumed to be coming out of the plane, and vertical bonds are used to indicate substituents above and below the 6-membered ring. In this slide, we are given the step-by-step process on how to convert a Fisher projection formula into a Howard projection formula. Okay, so let us zoom in. Step 1. So in step 1, we are given the Fisher projection formula of the glucose. In step 2, it says that all OH groups to the right in the Fisher projection formula appear below the ring, whereas the OH groups to the left appear above the ring. So if we will go back to step 1, you will see that I encircled OH groups using the red circles. Now, in step 2, you will see that these OH groups appear below the ring and they are still encircled in red. Again, let's go back to step 1. You can see that there is an OH group on the left side, right? And I encircled it using color blue. So in step 2, that OH group from the left side appears above the ring and encircled in color blue. Now let us look at step 3. There is a counterclockwise rotation of the groups attached to carbon 5, which will give the structure below step 3. Okay? But in order to understand that, we have to go back to step 2. There is an arrow there colored red. Okay, so what you can see, the direction of the arrow is rotating counterclockwise. So that red OH group, or that OH group highlighted in red, will replace the position of CH2OH on the right side. And that CH2OH now will be transferred and will replace the hydrogen atom above that carbon 5. Okay, and that hydrogen atom now is what you can see here. I encircled it in color green, will be positioned below carbon 5. Okay, so you just have to go back to step 2 and look at the direction of the arrows, which is rotating counterclockwise. Again, the OH group highlighted in red will replace the position of CH2OH, and the CH2OH will replace the hydrogen atoms above carbon 5, and that hydrogen atom will be transferred or positioned below carbon 5. Okay, that hydrogen there that I colored green. Okay, so let's look at carbon 4. I mean step 4. The OH group on the carbon 5 adds across the carbon double bond O. Right? So we have OH groups from in step 3. Okay? So that OH group now will add across the carbon double bond O. So in step 5, okay, the structure on the right side of step 5 and 6 will look like that after the OH group adds on carbon 5. Okay, so just look at the direction of the arrows. And it's in step 5. It mentions here that there will be 2 stereoisomers possible, or we call them anomers. Okay, so in step 6, it depends on how the ring closure occurs. So we have the alpha and the beta forms, and we call them anomers. So in the alpha, okay, look at that one. The O and the H are on opposite sides. Okay? And look at in beta form. The O and H are positioned on the same side. Okay? I hope you get it. So these are additional information on how we converted the Fisher projection into Haworth projection from this rules or step-by-step process. Okay? So you can read that by yourself later on, but I recommend this one since this is the step-by-step process already.